Today I'm going to be talking about some of my work. I'm an ecologist and primarily focused on coral reefs, and in particular what we need to do to keep coral reefs around for as long as we can. So I'm going to be talking about coral reef resilience and why in this recent project I did in explicitly incorporating scale into this long-standing question in the field revealed a pattern that nobody has been able to reveal before. That's super important. So hopefully this is a little bit motivating at the end of the day that we're doing this for a reason. So a little background, coral reefs worldwide are really threatened, and one of those threats includes marine heat waves that cause coral bleaching. So each of these white corals in this picture are not supposed to be that white, and this happens when the water gets too hot and stays too hot for too long, and that's happening increasingly with climate change. So what's happening there is actually at a molecular and physiological level. There's a breakdown in the symbiosis between corals and the symbiotic algae living in their tissues. So these are microscopic algae. You can see them where they're in high densities in this polyp. And so you can see how this is a scale issue because this is a process that's happening at a molecular chemical level within a polyp. Hundreds to thousands of polyps make up coral individuals, and then many individuals make up a reef. And these reefs span entire islands, or as big as the Great Barrier Reef. And so we have this scale problem. And much of what's been done for coral bleaching is actually at this level. So it's mainly chemistry. That is what we really know about coral bleaching, and not what explains these broader scale patterns, which are at the level at which the heat waves are occurring and the scale at which we can do something about it, kind of picking up on the last talk. And there can be a lot of spatial variability in bleaching. So this is an example from some recent work of mine. This is an island in the South Pacific called Moorea, which is off the coast of Tahiti in French Polynesia. And these data, this is an interpolation of point data from around the lagoons of Moorea, where these two types of coral live. And the darker colors represent the greater severity in bleaching. And so you can see there's huge spatial variability. And so that was kind of the motivation for the question of this research, which is whether we could explain this spatial variability in bleaching, given spatial variability in temperature, but also in pollution, a human influence that's occurring at the scale of this island. And so jumping straight into the results, this is what we found. We found an interaction between nutrients from pollution and heat stress, and interactions are complicated, so we'll break this down for a second. So we have low, moderate, and high heat stress. So if you just look at the intercepts of this, you can see the increasing effect of temperature, where there's a higher bleaching severity where there's hotter temperatures. Okay, we already knew that. What we didn't know is that at low temperatures, we could get just as bad of bleaching at high nutrients. So this is super important because effectively we're reducing the threshold at which bleaching occurs with a local human impact in light of this global human impact. And so that suggests that we need to both mitigate pollution at a local scale and climate change at a global scale to save coral reefs. So we better get to work. And okay, so now getting to the point of the talk at exactly 5 minutes is how did we get this answer when nobody's been able to find this before? This pattern's been shown to some extent in experiments, typically in the lab, not even in nature. And so we uncovered this at the scale of an entire island. And the way we were able to do that, I really believe, is because we incorporated scale into the analysis. So the data were at the scale of individual colonies, and there was over 8,000 of them. And as I mentioned at the beginning, really this bleaching really occurs at the scale of these individuals and their physiology. And so we needed to start there. And we incorporated predictors at that scale that we knew related to this process at that scale. So how deep the coral was in the water and how big it was. And then each of those individuals were nested within sites. So there could be many, anywhere from a few to 1,000 individuals at a site, where site's about 200 meters by 200 meters. And at that scale is the scale at which we had nutrients and heat stress varying. And so we were incorporating these predictors at this scale that we knew was related to the process. But also, we were able to incorporate this data at the individual level, taking into account variation. So not just means, we're not just aggregating. We're thinking about both the mean and the variance, and how that then relates to this hierarchy of scales. And so that's what allowed us to reveal this important pattern. So that's just reiterated here again. We matched the scale of the data collection with our understanding of the scale of the processes. And so I just wanted to hit that point a little bit harder based on some of the conversations we've had already, in that sometimes I think we're looking for the scale that that the process is occurring. Maybe we don't know. But in an example like this, in this ecological system, we have a pretty good idea of these scales to some extent, and so we can structure our questions around that. And it's just a different way of thinking about incorporating scale in analysis. I did want to point out, though, that potentially these scales are arbitrary. I decided them beforehand. And so maybe that's an issue, or maybe it's not, and it's a lot of the context in my mind for this was hierarchy theory, complex systems theory, and resilience theory, which don't necessarily translate directly onto geographical theory. And so I think that could be a fun point of conversation as well, and that's come up again today a few times. So what's next? We have a lot of cool data. So if anyone's interested, again, our data kind of starts here, but I have many colleagues who work at this molecular level, and so and then up to remote sensing as well, and we can get to really broad extents. And so we're working on kind of how we can get from one end to the other of this problem. Cool. Thank you. A really long